So, I'm going to talk today about I'm an ALS researcher and ALS clinician, right? And I think a lot lately about what does that really mean? What does it mean to be an ALS expert? I mean, can anyone really say they're an expert in ALS when we haven't been as successful as we'd like in figuring out ways to treat this disease? And, I think myself and a lot of really smart people that I know all over the place that are doing research on this disease, the way we're approaching it is changing. And it's changing because we are learning that what we all have to do is we all have to work together, and we have to leverage all of our resources, and we have to work with all of our patients and all of the pharma companies and all of the philanthropy, and everybody really has to figure out how to do this together. That it's a war on ALS, and if you're going to do a war on something, you've got to bring together all of your various resources. And the way that we're thinking about this disease and the way that we're trying to come up with new therapies is changing. So I'm not going to, do any sort of whirlwind tour of here's all the research and here's all the things. I'm going to talk about a couple treatments and a couple tools, but mostly just to illustrate how I really, really hope that that we're right on the crux of seeing a whole lot of new treatments and a whole lot of progress, and I hope I can convince you of that. So we'll briefly talk about how to diagnose ALS. I know this is a really savvy audience because I know, who takes care of you. And, but then, how do we think about a disease? And not just ALS, how do we think about any disease? And how do we try to find treatments? And why have we not done a good job? Why have so many treatments failed? What's wrong? How can we do better? And really most importantly, because I get this question a lot, is what's all that ice bucket money going to, right? So recent history of ALS, 1939, right, Lou Gehrig, this is when most people kind of learn about this disease. The first clue about at least one thing that might go wrong in ALS, this idea of something called glutamate excitotoxicity, late 1980s, early 1990s, there was this idea that there's this chemical glutamate that there's too much of, and maybe this is a bad thing, and maybe it's killing nerves. And interestingly enough, soon after those experiments were done, so actually sort of around the same time, but early 1990s, we had our first animal model, right? So this is a way that we do research. We have, a model, some way that we're able to create a disease in a lab, and then do tests on that and discover treatments. And the reason we had an animal model is because in 1993, the very first gene, the first genetic cause of ALS, SOD, was discovered. And certainly there have been a lot of genes discovered since then. 1995, Ryuzol, right? So look at that. Look how quickly that happened. That's something that doesn't happen as much anymore. But sort of figured out possible mechanism, got to testing it, and had this first drug. And I wasn't part of this community then, but I can imagine that back then there was a lot of optimism because look at this. We figured out something about this disease, and now we got this new treatment. And as Terry was just saying, if I gave this talk last week, I would tell you that it's the only treatment we still have, right? Quite remarkably, this past Friday, we got another one. We'll mention that in a moment. But this drug, Ryuzol, is an anti-glutamate drug, and so we had an idea, and we tried to target that idea. More recently, we've learned a lot of genes, but the gene that turns out to be the most common cause, genetic cause of ALS, the C9orf, was discovered more recently, in 2011. And it also causes dementia in some patients. And in 2014, this ice bucket challenge went crazy on viral media. And I don't think we've ever seen anything like this before. It was really remarkable. And I have patients from other countries who don't speak English and they're not ALS patients, and they never heard of ALS, and they never heard of Lou Gehrig, and they never heard of motor neuron disease. But you say ice bucket challenge, and they say, "Oh, my kids in school, they did that." And it really was unbelievable how much awareness and visibility it got. And then, most recent history, of course, we have our second drug. And I really hope and I really think that this is just the beginning, that we're going to see an accelerated, more drugs coming out more quickly because of, in some ways, the ways that we're doing things differently now. So certainly this is not something that I have to explain to people in this room, but it's important to think about what do we mean when we say ALS, right? Because what is this disease? And it really hasn't changed in a long time, right? ALS is a disease. It's a clinical diagnosis. It's a pattern recognition. It's not a lab test. You don't go to the doctor and the doctor does a test and says, oh, your test is positive for ALS. You have weakness. The weakness starts somewhere and the weakness spreads. And it doesn't involve all the other parts of the nervous system. It is disease of the motor system. And we still don't know what causes most cases of ALS, right? So we know that a portion of ALS has a family history, and we know some genes that cause some ALS. But in most cases, we don't know. We don't know what causes it, and we think there's a whole lot of different causes. This here is a picture of, it actually ends in 2015, but over the years, these are different genes that have been discovered that can cause ALS. And the size of these circles is supposed to mean how common that is. So this is C9orf in 2011, the most common cause. This is SOD, that first gene I mentioned. And you notice this curve, it's not a straight line, right? It's going woo, and it's kept doing that. So we keep learning more and more. We keep learning more genetic factors that are involved in this disease. It's, this is an age of big data and computers and analyzing everything. And despite that, though, we still don't know what causes ALS for most of our patients. So ALS is a clinical diagnosis, right? And so what does that mean? So what does a neurologist do? So when you go to a neurologist and something's wrong with you, the first thing that they're trying to do, they're not trying to say, do you have Parkinson's disease or ALS or MS or a tumor? What they're trying to do is say, where can I point my finger in your nervous system that is telling me that the problem is, right? How can I examine you and figure out from your exam, where's the problem? And then once I figure out where it is, then I can think about what are the kinds of diseases that affect that area. And that's what we mean when we say a clinical diagnosis. So I like history, I like medical history. This is a book from 1912. I like famous neurologists too, and people who wrote books. As far as I can tell, but maybe someone else knows differently, this dude's not famous. He was actually in like the lowest level of faculty, and low level faculty, we write these like case series and things because we're told to. But anyway, nothing's changed. This is 1912. But this is a drawing from this book. I like it. It's because it's a drawing of the motor system and the way the neurologist might think about where is the problem if you have a problem with weakness. So this is supposed to be a cartoon of a cut through the brain. And there's a part of the brain, of the surface of the brain, called the motor cortex. And your body's represented in it, your face, your arm, your leg. And these are all the parts of the brain that control the movements. And all those fibers, they come together. And they come together and they go through this deep area here called the striatum. And then they come through your brainstem, and they go down your spinal cord. And they go down your spinal cord in this tiny little package. And then eventually they touch another neuron that goes out and makes your muscles move, right? This is newer pictures, but they're no better. I mean, it's showing the same thing. But, here's that part of the brain, right? Here's your face, arm, leg. And the fibers come down, and they go through the brainstem, and then they come down, and then they go out, and they go and make your muscles move. This is a cross-section of the spinal cord. See all these little things and all these words? That's a lot of stuff, right? Spinal cords are small. I don't know if anyone... So your spinal cord is actually a little thinner than my pen here, and yet it's got all this different stuff in it. And the motor stuff is only in these tiny little areas, pardon my tremor. And yet somehow, as this disease spreads, it only involves the motor system. It doesn't involve any of these other regions. So this is my cartoon. I'm more towards the 1912 guy. This is my drawing of the motor system. So we got brain, we got spinal cord, right? In the simplest sense, you have neurons up in your brain that the cell bodies are up here, and they come down your spinal cord, and they touch other ones, and these other ones go out, and they touch your muscles. That's M. When you look at someone's muscle, there's big M's on them, and they make the muscles move. And ALS is a disease of weakness, and you can have weakness because of a problem anywhere along here, right? So you might know somebody who's had a stroke. That's a problem up in the brain, and they can't move some part of their body. Someone can have an injury to their spinal cord, or can have arthritis in their spine. They can get weak. Someone can have a problem with this nerve coming out of the spinal cord. That's neuropathy, like diabetes, things like that, where the nerve connects to the muscle, diseases like myasthenia gravis. You can have muscle diseases, myositis, muscular dystrophy. So a problem anywhere along here can make you weak. When a neurologist is examining a patient, if I draw a line through this picture right here, if the problem's on this side of the picture, the brain, spinal cord, you see certain things on exam. Those are upper motor neuron signs. I don't think I'm saying anything that people in this room probably don't know, but I think it's important to lay this groundwork. So, jumpy reflexes, spasticity, emotional lability, those are problems in this side of the picture. If the problem is in this side of the picture, muscle mass loss, weakness, twitchy muscles, loss of reflexes. ALS is a unique disease, and the way we make the clinical diagnosis is that we get problems here and here, and we get them gradually and progressively, and they start in one part of the body and they spread. And so while we don't have a test, there really is no other disease that does that. And that's really how the diagnosis of ALS is made, that it's this combination of these upper and lower motor neuron signs that start somewhere and gradually spread out and don't involve the rest of the nervous system. So the localization is what leads us to the diagnosis. And then how do we think about what's wrong? Well, I mentioned the very first thing we knew was this idea about glutamate, right? So the first idea is that glutamate might be involved, or excitotoxicity. It actually came from some environmental studies. There's this Lathyrus sativus, this chickling pea, which in certain Asian countries is in the diet. And people who eat a whole lot of it, it contains this chemical here, which looks a lot like a chemical in our body, glutamate. And they got this disease that sort of looks like ALS. And so, people thought, that's interesting. They got some chemical, the chemical looks like something that's in our brain, and they get a disease that looks like ALS. So what is glutamate? This is the famous, the most renowned neurology textbook that you guys all know of, right? Wikipedia. Everyone has a copy on their bookshelf right next to the Encyclopedia Britannica. And so glutamate, is a neurotransmitter, an excitatory neurotransmitter. It's the way nerves talk to each other. You have one nerve, it squirts it out, and it works on the other nerve. And the interesting thing is, if there's too much of it, if it's too excited, that's what can cause, presumably, some nerves to die. So it gets squirted out, and then it gets taken back up, and it gets taken back up. And this little pictures, I guess, that's how small it would be, but through things called glutamate transporters. And so it gets recycled. And also these other cells called astrocytes have glutamate transporters on this green thing to sort of get rid of that extra glutamate so that after it signals, it doesn't do its thing. Glutamate's not just in the motor system. Glutamate is everywhere in your nervous system. It's the excitatory neurotransmitter. Glutamate, by the way, when we say it's everywhere, it's what makes mushrooms make your steak taste better, right? MSG, it truly is. It's not just in the motor system, it's all around our nervous system. And by the way, you can learn a lot from good eats. So is there maybe too much glutamate in ALS? So Jeff Rothstein at Hopkins was the first person to think about this, and what he did was he took spinal fluid, the fluid that bathes your brain and your spinal cord, from patients and from people who don't have ALS, because this is how we think about what might be different, and said, can I see anything different? And so measuring in that spinal fluid, these bars here, so this is glutamate in spinal fluid, and this is another aspartate related to glutamate, but compared to people without ALS, there's a whole lot of glutamate in the spinal fluid. And those transporters, those things that take up the extra glutamate, those were missing. And this is the motor cortex, and this is the spinal cord in the patients with ALS. So something was wrong where ALS patients had too much glutamate and they weren't doing a good job of recycling it. And when they looked at tissues from those mice, those SOD mice, that mouse model that we had, and also from patients, so this is a control patient staining for those glutamate transporters. This dark stuff is the stain, and it's just not here in the ALS patients. And specifically, we have all this alphabet soup in science, but this one here, this thing called E2, which is on these cells next to the neurons. So controls, you've got a whole lot of these glutamate transporters, and in most ALS patients, not all, it was missing. So this is where this idea came from. And so how would you make a drug? How could you target that? Well, that's actually where Riluzole first came from, right? Because what Riluzole does is it decreases the release of glutamate, so it makes you have less. And in fact, Riluzole works, right? It doesn't work well, it works a little bit, but it always works. It slows this disease down. It doesn't matter what kind of ALS you have, genetic, fast, slow, in your arm, it actually does work. And so these kinds of plots are the sort of things where we see where this is patients not getting it and the patients getting Riluzole, they live longer, okay? And we've since learned that if you started earlier, you live even longer than that. So what if you were how about trying to make there be more of those transporters? Rather than decreasing the release, what if you could somehow speed up or increase those things that are supposed to be getting rid of it? And so they screened about 1,000 different drugs, looking for things that would do that. And they found that certain antibiotics, in particular ceftriaxone, you guys remember ceftriaxone? There was a lot of excitement a few years ago, because ceftriaxone in mice made glutamate uptake happen more efficiently and better, and made the mice live longer. And in a phase 2 trial in people, it also helped. It prolonged survival. And then this big phase 3 trial that we all had a lot of excitement about, and it failed. It didn't do anything. So what happened? Why is there a problem here? What's the disconnect? Why did something work in a small trial, work in mice, and not work in our patients? So we're going to get back to that. Since that time, we've learned a lot about this disease. We've learned there's lots of cells involved, right? You see these pictures where there's 47, look at that mess, right? These are all different mechanisms. You know, there's neurons and other cells that are supposedly involved in creating this disease. And so where does this come from? Here's a list of just some of them. Some of the things that we're targeting, some of the things that we have discovered possibly are in some kinds of ALS messed up. You'll notice these are things, it's not just your motor system that uses this, these are processes that every cell in your body uses. Mitochondria are the energy for all of your cells, right? Cytoskeleton is the structure of all of your cells. These are really general cellular mechanisms. So where did this come from? Why do we get this? So this is our house. So 3 years ago, we moved into a house that's way up in the country, and it's this old stone house that was built in like 1943. And when we moved in, it was a disaster. It had been vacant for, 40 years? Overgrown with weeds, basements filled with mold, everything was broken, nothing worked. Our parents said, "You're crazy, what's wrong with you? Why are you buying this house?" The only reason we could afford the house is because it was a mess. Other than my wife over here, Kimberly, shaking her head. And our parents tried so hard to talk us out of it. And we really worked our butts off and got this house into great shape and fixed everything. And we're like, yes, we love it, it's ours. And then that was that year we had the really bad winter, right? And that winter we discovered something, which was that this house was the draftiest place I've ever been. And if there was a cold breeze blowing outside, there was a cold breeze blowing inside. It was freezing. Now, if you were comparing my house to the house next door and saying, what's different? If you didn't know what made a house cold, right, your patient and your control, the first thing you might notice if you were just looking at a microscope through a cut through my house is there's a whole lot of ash in our fireplaces, right, because we were burning a lot of fires. You know, interestingly enough, some of these things that in ALS that are being investigated are accumulations of stuff in cells. And we never really, in some cases we know that the stuff does something bad, but you always wonder, is it doing something or is it ash in the fireplace? Because I'll tell you what, if you took the ash out of my fireplace, that's not a treatment for my house being cold. It wouldn't help. Now let's say you understood something about how a fireplace worked, and so you knew, oh, that ash is there because you're burning a lot of fires, right? So maybe you should burn more fires. Well, you know what we learned that first year? If you burn a lot of fires, it makes you warm in that little area around the fireplace, but it actually makes your house a lot colder because it sucks all the drafty air in from everywhere else, right? Cools down your house. We have oil heat. This is me running out in the middle of the night, not knowing anything about oil heat because we're freezing, and we discover this is a stopper. It's supposed to be up here showing that there's oil. This is our empty oil tank, okay? We burn way more oil than our neighbors, way more. So if you concluded that, oh, the problem is you need to burn less oil, no, that's not gonna help, okay? This is our furnace. It's a fine furnace. It's probably maybe the same furnace as my neighbor has, but guess what? Our furnace breaks down more frequently. Our furnace needs more service than the neighbor's furnace. Is the problem the furnace? No, but it's working a lot harder to keep the house warm. So what's the gene defect in our house? What's the genetic problem, right? Because if you knew what the problem was, you'd like to address it. The problem is we got these windows from the 1940s that have these steel sashes and single panes, and it's a stone house, and there's not much we can do about it, and everything's freezing, right? Ideally, you would want to address this. You would want to replace these windows, the genetic defect, fix the defect. It's hard to do. It's hard to do gene therapy. This is my father-in-law. So our parents would come visit, and they would like turn the heat up, right, the thermostat up, because they were always cold. And we were burning, $24 of oil every day, and it was crazy. And we'd follow them around from room to room, turning it back down. And so for Christmas one year, we bought my father-in-law this, infomercial footy pajama thing, right? And besides the awesome side effect of the treatment that made him look really silly, it's actually a good treatment. Okay. Now, does this address the problem with my windows? No. But does it make him not feel cold? Yes. Okay. So, what is this? This is tirasemtiv. Is anyone in this room part of this trial? So you don't need a treatment to necessarily target your disease mechanism. Tirasemtiv is a drug that increases the efficiency of the contraction in muscle fibers, not specific to ALS, but it's like the footy pajamas, right? And so, we're hoping that this is going to be a good treatment. Now, so what's the point? The point is that our body has these interrelated pathways that try to compensate for an abnormality. And the thing about ALS is there's probably many different causes. And we need to know if we build a treatment based on a model of a mouse that has one gene mutation, does that necessarily mean that that treatment's going to work? How do we know which patients it's going to work in? Patients that progress fast, slow, patients that have different sorts of abnormalities. We also don't quite still understand how all these general cell mechanisms can lead to this one very specific disease. But we know that something's wrong, that there's a disconnect, right? Because we have these data in our models, and when we take them to patients, they don't do well. This bullseye here, this is all the drugs, and this is way out of date now even. This is all like phase 1, 2, 3, 4. So not many drugs are making it through, okay, even though they work in our labs. Some drugs have actually sped up the disease. We've learned something from these drugs. Some of the drugs that sped up the disease were ones that caused a side effect of weight loss, and that was one of the first hints that maintaining your weight slows down ALS. But while the models are very important to study what might be wrong in ALS, what we've learned is that we really need patient-based tools, things that we can go and say, okay, this is fine in the model, but are we sure that this is really happening in our patients? And maybe it's only happening in some of our patients, and how do we figure out who those patients are? And so I'm going to talk a little bit about each of these things, but one of the things, I'm not sure if anyone in this room has done this, but we can take skin cells from our patients, and now we actually take blood, and we can turn those into stem cells, what are called iPS cells. And then those stem cells can get turned into different types of cells in your body, like neurons. And this is different. This isn't the stem cells for treating, right? This is stem cells to have as a laboratory tool, because you can do this from a patient with ALS, or a particular kind of ALS, or 27 kinds of ALS. And you can compare those cells to cells from somebody who doesn't have ALS. And then you can throw drugs at them, and you can say, can I make the one from ALS look like the one that's not from ALS? And in fact, this is what we do. And this is just, so for C9, this very recently discovered gene, without saying going through this data, these columns are showing that the pictures from stem cells and the pictures from patients' tissues, they do the same thing. And so we can make something in a dish that looks like a patient, and theoretically you could do a clinical trial in a dish. And if you know a gene, you know, like I said, with my windows, the best thing would be to turn off the gene if the gene's doing something bad. 10, 15 years ago, gene therapy was sort of science fiction. You know, it's actually not science fiction anymore, right? And one of the ways we do this is by something called antisense, which turns off these genes, and we figured that out by doing it in this dish first, and realizing that we could turn these genes off in the dish. Antisense has been around for a bit. It's used in, so this is for myotonic dystrophy, but the idea is that you've got these little tiny pieces of things that look like RNA and DNA, and they go bind to it, and they keep the bad thing from doing what it's supposed to do. And so can we do this in ALS? Unbelievably, we can. So this was, 5 years ago, this was the first gene therapy trial in a neurologic disease, and it was in our ALS patients with SOD. This was using an antisense to SOD. This trial wasn't designed to make anyone better. It was designed to prove to the FDA that this was safe and that we weren't going to make things worse. And I have to tell you, this was the most remarkable experience for me to see what our patients were willing to go through to help put this forward, because patients came in and they got admitted to the hospital and they got a giant catheter stuck in their back and they got an infusion for a day and a half, and every 15, 20 minutes they were getting blood samples and urine samples, and it was just remarkable, and they were doing this enthusiastically and doing this for the good of the community, and it was really just a moving thing. And this has now moved on. There's a second version of this that's being tested. But this has led to other things, right? Have you heard of Spinraza? So Spinraza was the Christmas miracle for this childhood disease called SMA, which is a fatal, horrible muscle disease. It's related to ALS. And an antisense treatment, in fact, works. And so this is our proof of principle that gives us hope that this sort of thing is going to work in ALS. There's now trials being planned for C9 ALS, more for SOD. So if you know the target, if you know the gene, you might be able to do something. But what if you don't know, right? Most patients, we don't know what's causing. Or what if it's not that the gene does something toxic, but you're missing something? You can't really do it the same way. What do we do? How can we fix this problem with our animal models? So this is where this idea came, that we really need to be validating what we find in the lab in human tissues. And you guys might have heard there's things called brain banks, right? Alzheimer's research banks. These have been around for a long time. We've never had a tissue bank for ALS, and our needs are different than these other diseases, because these other diseases have changes throughout the brain. ALS, it's in a very specific area, that motor strip, right? The spinal cord. Alzheimer's, Parkinson's, they don't care so much about the spinal cord. Doesn't affect it. So we really didn't have the tools and the resources, and so that's why we formed this tissue pool. And so what we did was we took 5 centers that all were good at brain banking. We got together and we harmonized. We figured out how could we all do things the same way? How could we standardize? And how could we make this the best possible for ALS research? So that every person who donates, because it's really the ultimate donation, so that if a person donates their body to ALS research, that tissue is going to be used in 200 different experiments, not one experiment in one lab at one university, 2,000 different experiments. These samples and all the data with them are provided free of charge to anyone in the ALS research community, academic, industry, anyone. This project is funded by Target ALS. Target ALS has a very unique marching orders. The marching orders are, they're not funding a lab to do something or a lab over here to do something. They're funding consortia, and they want people to work together. And not just consortia, but consortia that make industry work with academics and vice versa. And funding these research cores to make tools that help everyone. This is on the website. This is this very simple idea. Oh, we've got these centers. They're all going to do things the same way. We're going to have a review committee, and the review committee is going to decide if you get tissue. I'll tell you what, even this was too complicated because we didn't want to slow anything down. So we no longer have a review committee. It's just me. And everything gets done on a rolling basis. And the reason is, like I said, if nothing is working, who am I to review and say that your idea is a bad idea or your idea is a bad idea because I don't have something better, right? So what we're reviewing is not the science. We're reviewing, can they do the experiments? Do their experiments work? And have they justified for the amount of tissue that they're requesting? And beyond that, that's it. We provide these samples. Everything is logged in a huge electronic database run by Niels and Neurobank, which Terry runs. And we have an online forum. Researchers can go online and look up and say, oh, I'm interested in this kind of ALS, and these samples processed this way, and what do you have? Every case we harmonize across all these sites. They all get the same pathologists looking at them the same way, making the same slides. We generated all these checkboxes. These are ways for pathologists to describe what they see under the microscope in a standardized way and make that into data that can help. So if you're a researcher and you're interested in a certain kind of cell or a certain problem, you can say, give me tissue that has a whole lot of that, or give me tissue that has a little bit of that. We now have made all those slides. There's a website, and anybody can go, and you can search by brain regions or type of stain or type of cells, and you can find these images, and you can do image analysis, or you can just decide, give me the samples that go along with those images. And all of this, again, not just for the samples that somebody requests, for this entire bank are available to everyone. People kept requesting these samples to do genetics, right? To say, oh, I want to look at my gene that I'm interested in. And we kept sending out samples over and over to do the same thing, and we realized this is not efficient. What if we just did all the genetics at once and did it the right way, and then let's make all that data just available to everybody right away? Not just genetics, but looking at RNA, looking at how are different proteins expressed in different regions of these brains. And so part of so funded by that ice bucket money, the New York Genome Center, and by the way, let's not do this because when we were giving out samples, one lab's good at doing it, one lab's not so good, one lab makes a mistake in this. We don't want to waste this resource. And so now we have all of this data for all of these cases, and not just for our Target ALS, but this has sort of been growing where other big data efforts have been giving this data as well. And it's available to anybody that wants to use it, okay? All free of charge. So remember that picture with the 5 things coming together? It's actually become much more complicated now. This is kind of the workflow from a single autopsy and what happens. And this isn't even the side where we then give the stuff out to the provider, which is very complicated because it comes from different sources and all sorts of things. But it works. These are just some of these projects that the tissues have been used for. These are not, as I said, maybe the typical things you hear about, and that's because who knows what's going to work? And so we're open to let anyone try what they want to try. We have, and since this started, we've given out tissue over 90 times, hundreds of samples each time, so we're talking thousands and thousands of pieces of tissue. Been used for more than 60 projects. We provided letters of corroboration to help young investigators get grants. We have, and anytime we can, foster collaboration between, so if 3 people request stuff from us to do something in a similar area, we say, hey, you want to talk to them and let us put you in contact, and maybe you guys can work together. One of those I just want to highlight, this endogenous retrovirus project, which maybe some people have heard about. So Avi Nath, who's the clinical director at NINDS, found this really fascinating thing. So we've got this stuff in our bodies, it's called junk DNA sometimes, but it's actually, it's remnants of ancient viruses millions of years ago that infected our ancestors millions of years ago. And little bits of their DNA got stuck in our cells. And they just sit there quietly and nobody thought they did anything. But it turns out that some of them get activated in some diseases. And these viruses, they're a lot like HIV, they're retroviruses. And so when they get activated, they sort of turn on their own machinery. And we found, using the human tissues, that in fact, this one called HERV-K, human endogenous retrovirus K, is activated in some ALS patients. Retroviruses use a particular enzyme called reverse transcriptase. You can measure it in somebody's blood. You can potentially, when somebody is an HIV patient getting treated for their HIV, their doctors follow that level to see how their treatment's doing. We can measure it in some patients. Some ALS patients have elevated levels of this reverse transcriptase. Dr. Nathan, I won't play it, but made a mouse model by taking this little tiny piece of this one protein, not even the whole virus, just one protein, and expressing it in mice, and the mice get ALS. So this ancient viral infection protein, you put it in a mouse, and it is actually a better looking mouse model of ALS than almost any model that we have. And the remarkable thing is that because of this collaborative atmosphere, people took this and they ran with it. So pharma companies that were making antiretrovirals for HIV now say, hey, wait a minute, maybe my drugs can work for some patients with ALS. Infectious disease doctors who were interested in these kinds of viruses suddenly are interested in ALS. We just had the second international conference on human endogenous retroviruses, an entire third of this conference devoted to ALS. This is Avi. It's kind of hilarious. They made me chair this thing, but I learned everything I know about this from him, so it was kind of embarrassing that, it's this science fiction-y, fascinating story that's now been taken. I'll tell you, there's a trial right now in Australia using antiretrovirals to try and treat some patients with ALS, and we're going to have one in this country soon. And it grew out of this collaborative atmosphere of letting everyone have access to all of this data, all of these ideas. So we have better tools, we have better things to use. Trials still fail, right? They fail a lot. The ceftriaxone trial, why did it fail? Right? Did it fail because the idea was wrong? Because it was just wrong, and something was wrong about the early data? Or maybe the drug didn't get there. Maybe the drug didn't get across the blood-brain barrier, which has to do that to get to where it needs to work. Or maybe it got there, but maybe it didn't do the thing it was supposed to do, fix that glutamate, right? And so we don't know. We have a huge trial. We wasted not wasted, but a lot of money. And at the end of it, we still don't know that answer. And we wish we did. We wish we had a way to track, did the drug have an effect? And because ALS, as we said, it's a clinical diagnosis, and patients are very different, right? Some patients are slow, some are fast, some have different parts of their body, different things involved. It's difficult to run a clinical trial and know whether, and at the end of it, if it doesn't work, why didn't it work? And so there's been this big push to have what are called biomarkers, right? And this, again, has been very largely funded by the ice bucket money. So biomarkers, any measurement, blood, urine, anything that you can measure that can track the disease process. And the idea here is that pharma companies, they don't want to fund ALS research anymore. Can you blame them? I mean, not all of them, but it's hard to say, I'm going to give all this money to fund a trial, and then that trial doesn't produce something. We need biomarkers. We need to know, is the drug getting to where it's supposed to go and having the effect it's supposed to have to tell whether it worked? There's different kinds of biomarkers. So if a drug is targeting one of those mechanisms, let's say inflammation, you would like to know which of your patients have a whole lot of inflammation. If it's targeting a particular problem with cells, is there a way to tell which patients have that? Because those are the patients you'd like to enroll in that trial, right? The ones that might benefit from it. There's biomarkers that can track whether a drug has a response on the disease. So this is the problem, though, or one problem. So any measurement that tracks, so this is this website, if anyone's ever seen this guy, Tyler Vegan, he wrote a computer program that mines publicly available data sets of all sorts of stuff and finds correlations, right? You can go and play with it, you can actually do your own. So this is the divorce rate in Maine over this decade from the US Census compared to the per capita consumption of margarine across the entire US. And that's from the USDA for that same decade, right? And it's unbelievable, 0.99, like, oh my, I wish my data in the lab looked like that. Okay, clearly, people getting divorced in Maine is because we're eating too much margarine, right? This one I like even more. So this is the per capita consumption of cheese in the whole US, and the number of people who die by becoming tangled in their bed sheets. Okay, now, I have to say, and again, an unbelievable correlation. Whether or not these two things are related, it does bother me that the number of people who die by becoming tangled in their bed sheets is increasing. Like, is that a bed sheet problem? Is it because we're not teaching our kids to make their beds? I don't know. So in this age of big data and collecting everything from everybody, you're going to find correlations. And so, you have to just be careful. As I say, a biomarker doesn't necessarily have to be involved in a disease process. It tracks the disease process. But it's better if it does. And so now we have all these tissues, and now we have all these needs for biomarkers. And so the ALS Association, so the pharma companies were saying, what we really need is not just the tissues, we need fluids to go along. We need to know, what does the tissue look like, what do the fluids look like? And so our effort has now spurred more investment to add biofluids onto our core. Those biofluids go into the Neurobank repository, which Terry runs, everybody coming together, everybody harmonizing, doing things the same way. We also can use imaging, right? So you can look at a change in an MRI scan over time. Maybe you could do an image study that just looked at the mechanism your drug's supposed to be acting on and see if it's changing. Terry mentioned PET imaging. So PET imaging is a way, PET imaging is most commonly used. You inject a radioactive something into someone and then you see where it goes and then you measure that radioactivity. And the way it's used in medicine usually is you put radioactive sugar into somebody, right? And all the cells in your body use sugar, glucose. And if cells are using too much sugar, well, tumor cells use too much sugar, or inflammation uses too much sugar. Dead cells don't use any. And so it's a way that people look at different metabolism in different areas of the body. But you can make a PET ligand from anything. You can make anything radioactive. You can make, and then you can track a change in that thing in the body. So this was this trial that actually I just finished at Hopkins, again funded by Ice Bucket money. And so mGluR5, what's this? Why do we have to have so many letters and numbers? It's impossible to keep track. Remember that thing glutamate again that acts everywhere and does everything? Glutamate acts on all kinds of different classes of cells and molecules. This is just one of them. And it's one that we really didn't think was involved in ALS, but we had information from human tissues that as ALS progresses, it went up. Now, we didn't know if it was causing a problem necessarily or if it just happened to track with it. There was some data in those same mice that if you inhibited this thing, maybe they lived a little longer, but we actually didn't think this was very good data, and it wasn't why we chose this molecule. We chose this molecule for a simple reason. It was already available, already being used, already safe in people. It was being used for some psychiatric disorders. And so we knew it was safe, and we wanted to learn how to do PET studies in people. And we had some evidence that maybe it tracks with disease, but we had to figure out how to do this because, for example, that ceftriaxone trial, we're going to have a PET marker for the thing it was supposed to be affecting, right? Once that's available, we want to know how to do that trial, how to do the trial in ALS patients. So they come in, they get a screening visit, they get an MRI, because the PET scans are sort of low resolution, they look like old video games from a while ago, and the MRI helps figure out where you are, and then they get a PET, and you get these cool face masks made that sort of look like Phantom of the Opera, that's like your own mask, and you get a radio line put in, or at least used to, not anymore. And so we've done this, we did 20 ALS patients, or actually 18 ALS patients and 18 control patients. Originally, we had a lot of trouble recruiting patients. We had a lot of trouble getting people to do this, and mostly because it's not a treatment, right? Even though this would help us do trials better. Patients were understandably worried that, if I'm in one trial, what if a treatment trial comes along, and is that a problem? People don't like getting stuck with art lines, I don't blame them. And also this need to lie flat, and this is part of what we had to figure out. If you're going to do an imaging study in an ALS patient to track a treatment or a disease, that only works as long as the patient's able to lie flat, and as breathing is involved, that's a problem. So this is the kind of thing we have to overcome. We need to figure out how to do all these different visits. And this was an interesting thing that we hadn't anticipated. I had no idea how many of my ALS patients smoke marijuana. I'm not making any judgment, okay, but it's a huge percentage. And everybody was failing the radiology department's tox screens. For controls, we had a problem because their controls were usually much younger. We had to find a way to get older controls. We fixed all these things. We got rid of the art lines. We figured out how to combine visits. We got spouses to be controls. We gave people pictures of their brains, which they loved. It's kind of cool to see your brain image too. And this is from early on in the trial when we had just done 9 patients, and I think we had 6 controls. This is looking at healthy, this is looking at ALS. And this is looking at the motor cortex, that area that controls your brain. This here, see this bright stuff? This is this deep area of the brain, the caudate, and it's called the striatum. And we ignored this because it's high in the healthy subjects, it's high in ALS, it's even higher in ALS, much higher. But we don't really think it's involved in ALS, and so we said, that's artifact. We didn't pay attention. For those first 9 patients, this is in their ALS-FRS, how severe their disease is, and how much of that stuff was in their motor cortex. And we're like, hey, look at that, it sort of correlates, this may be going to work. The study's done now, we're analyzing the rest of the data. I don't have it today, but I'll just tell you that we found some very exciting things. While we were doing this study, someone took this same PET ligand and did it in those mice, those ALS mice, okay? And again, so white boxes are normal mice, gray boxes are mild ALS, black boxes are severe ALS, and this is the motor cortex. And so just like in our patients, as they got ALS and it went up. So maybe this is something that's going to track with disease. Strange thing happened though in this study they published while we were doing this. See this thing over here, striatum, this column here? This is that deep thing that I was ignoring and saying, oh, that's an artifact. And look at that, it's actually higher and increases even more, just like in our patients. So maybe not an artifact, and I'll just tell you. So this is that picture, right? Remember that drawing I showed you from 1912? So this area here, this is that bottleneck, right? This is where all those motor fibers are going through. Very recently, some other groups have shown with other imaging that that same area, this is now cut a different way, but this head of the caudate, what's called the ventral striatum, is changed both in shape and size and the connections that go around it in very early ALS and FTD, and I hope to be able to tell you more about this soon because I think it's going to open up a new way of thinking about some patients with ALS. So we have all these tools, we have all these biomarkers, we have all these new drugs. Still, it's like pot shots, right? Don't you feel that way when you go online and try to look it up, that there's like 47 things going on and you can't figure out what, how are we deciding what to do next or what's getting done or are they using the same strategies? And so another big thing that happened with the ice bucket money was this idea that we were going to get together hundreds of people, clinicians, researchers, patients, and we were going to make this ALS guidance. And I'm guessing that some people in this room were involved with this. We were going to make a document that could go to the FDA about how to do trials in ALS, how to standardize things. We were going to tell them, you need to include biomarkers in your trials so we know what happens. You need to help develop biomarkers. This had different working groups. I was actually one of the people who helped write the biomarker part of it, but there were several groups. And we needed to involve the patients in this. We needed to make this patient-centric, that you guys were telling us what we needed to do to make things work better and work faster and help us get to treatments faster. And all these people did this. This was hundreds and hundreds and hundreds of hours of work, and we all gladly volunteered and did it because we really want things to work better. We want to have a roadmap. About 2 months ago, the pharma company said, what we really want is longitudinal biofluids to go with your tissue and all this other stuff. And we said, that's great. Pharma companies usually have not funded biomarker studies because it's not, they want to look into their drugs. And so we got them all to come together, and not them all, but about 15 different companies and some organizations involved in ALS research, and sit around a table and say, hey, if we're going to design from the ground up a longitudinal, a way to find biomarkers and make them clinical trial ready, because we have a lot of potential biomarkers, but they have to be validated, can we all do this together? And can we do it together pre-competitively, meaning we all work together, all the pharma companies, all industry, all academic labs, and we do this as a combined project, and nobody owns it, and it's for the good of everybody and for the good of the community to get these tools that we all need, right? And they said, yeah, thumbs up. And so then after this meeting, I sent out a questionnaire to all these companies, an extensively long questionnaire detailing exactly what they were thinking about all these things. Where should this stuff be done? Which labs? What do we need to be measuring? What are the things that they're interested in? And we've now pulled all that data, and actually on Mother's Day, we're having a meeting where we're sitting back down and going over all of that together and getting everyone to agree that this is in everyone's best interest, that this is done, no intellectual property, no who owns the data, all of these players coming together for everybody, because we need to. We need to have these tools, we need to make things work faster. So the main point is that I think more than ever before, there's this harmonization going on. There's this figuring out, rather than having 27 people doing things differently, everybody does things still a little differently, but what are the parts we can do similar? How can we all work together? How can we all make everything available, play in the same sandbox, right? And use every resource available and leverage them more efficiently. And this is why I personally am more excited. And by the way, this also involves the FDA and hopefully accelerating and the pressure that patients and patient advocacy groups have put on them, I think played a role in what just happened a couple days ago, and I think it's going to keep happening, at least I hope that it does. So the interesting thing about a talk like this is an acknowledgement slide is kind of hard, right? Because it's everybody. So thank you for letting me come and talk to you, but it really is everybody, and it's everybody that's in this room too, because more and more we are depending, in an age when everybody is connected and, both on the internet and this information and ideas, and advocacy groups, it's more than ever, we're finding ways that it's not about who did the experiment or what was done in one lab. It's about everybody playing in the sandbox together and figuring out how we can do this quickly. Because, you know, I don't want to be an ALS expert if I can't give something to my patients. And I know a whole lot of really smart people all over the place and really smart patients who all have something to contribute. And so putting this infrastructure in place lets us harness everything together. So thank you.